I'm excited to introduce our second speaker today, Riccardo De La Peña. And I haven't been able to meet Riccardo, but I'm a huge fan of his work. And you know, he is currently at Stanford working in Elizabeth Satley's lab. But before going to Stanford and starting his PhD studies, he did his Bachelor's of Science in Chemical Engineering at the University of Texas in Austin. There he got some research experience working in the characterization of RNA structures. And so he's worked in RNA, and now he's come into this plant protein world where he's done a lot of beautiful work in the Satley lab. And so Riccardo, we're looking forward to your talk today. Hopefully we'll see some awesome plant natural products and learn more about their chemistry.
Thank you for the introduction, Allison. And thank you all for the invite. So today I'm going to be talking about, I guess, pathway discovery and metabolic engineering of complex plant diterpenes. So I think I wanted to start by just showing some valuable plant terpenes that have widespread industrial use. So I guess most of us are probably familiar with these products. I mean, digoxin, artemisinin, and Taxol are clinically used and very, very important molecules. But I think something that I want to highlight of commonalities of plant terpenoids is that a lot of them are very, very complex. So here I'm showing a different example. This is an antifungal molecule called adenosine from oats. And I kind of want to highlight some of the moieties in the terpenoid scaffold that make it a really, really hard target for chemical synthesis. So we have glycosyltransferases acting on the scaffold, a bunch of oxidases, and even acyltransferases. And if you add to that the stereocenters that the molecule actually has, you can start thinking about how impractical it would be to try to make this compound through chemical synthesis. So our alternatives usually then involve using the direct access of a terpenoid from the plant source. But here I want to highlight that this is sometimes a problem. So here I'm showing two ends of the spectrum. So on the left, I'm highlighting steviol glycosides, which most of us might be familiar as stevia, or the food sweetener, artificial food sweetener, low calorie. And this one accumulates at a really, really high level in the natural plant. So we're talking about anywhere between 4 to 25% of the dry weight of the leaf of the plant is just this molecule, which is just completely insane, or that's how I see it. But in the other end of the spectrum, we have, for instance, malolactones, which I'm going to be focusing my talk on. And these are allelopathic molecules that barely accumulate in the plant. So with that said, I think the key point that I want to take from this talk is that the utility of a terpenoid is limited without the elucidation of its complete biosynthetic pathway. So as I just mentioned earlier, I'm going to highlight, I guess, the chemistry and the biology of the rice malolactones. So I just mentioned that malolactones are allelopathic. So what is allelopathicity? So you got to remember that plants don't move around. They don't have an immune system. So they rely on usually chemical compounds for defense purposes. So in this case, allelopathicity occurs when plant A is near plant B, and plant A detects that plant B is close to it, and it exudes some sort of chemical compound to inhibit the growth, the germination, or the development of plant B and get some sort of evolutionary advantage. So in this case, plant A gets to grow big and large and likely sprout seeds, while plant B remains kind of stunned. So the malolactones are kind of the key examples of allelopathic molecules in plants. So there's been a lot of work in the characterization of the malolactone biosynthetic pathway. So when we started this project, I guess here I'm highlighting in blue everything that had been characterized previously. So these have been efforts that started since 2004. But there were still a few moieties that were left to be characterized, right? So we still didn't know how the lactone ring was made, or that last step, which would be the formation of that hemiacetal ring. And I guess doing pathway discovery is something that we do a lot in our lab, and we do it using three main things. We use transcriptomics, then to highlight, or well, not highlight, to, I guess, get at enzymes or genes that are likely involved in the pathway. And then we test them in a plant heterologous host. And then we do a lot of metabolomics to see which genes are active in our pathway of interest. So I guess I'm highlighting the way that we test our enzymes, which is, I guess, a little unusual. So we don't use a microbial host, we use the plant host for discovery and engineering of pathways. The reason why we do so is simply because utilizing a plant host will likely increase our chances that a protein of interest will likely fold properly, it will localize to the right organelles, and it just makes our life a lot easier. And here I'm kind of highlighting two things that you can do. You can do pathway discovery at a small scale, which would be just infiltrating one single leaf with agrobacterium. Agrobacterium, essentially, it's the pathogen that carries our gene of interest and introduces it to the tobacco leaf. So we can do that at a small scale, but we can also just essentially transform a ton of plants at one time and scale up and get large isolation of a lot of the product of interest for structural characterization or for use in whichever way we need. So I guess when we started, all of these enzymes had been characterized previously in vitro in expression of microbial hosts. So we thought, like, let's just start with what we know and let's test it in our plant heterologous system. And when we first express the terpene synthesis, we were able to see production of the syn-pyrimidine, which is a diterpene scaffold. However, when we added the first P450, we were no longer able to observe any product which would be essentially different than what people have previously characterized. Oh, sorry. Yeah, we weren't able to see the product. So we were wondering, why was this the case? And we thought about something that we typically do, which would be just try to boost the yields to see if it was just a problem of essentially not having enough precursor. And I guess working in a plant heterologous host allows you to do, I guess, kind of cool things. So here's a model for a plant cell, and I'm working on the terpenoid pathway. So I want to remind you that plants have two different terpenoid pathways that are distinctly localized. So we have the DXP pathway in the chloroplast that plants typically use to make diterpenoids or carotenoids. And then we have the cytosolic mevalonate pathway that plants typically use to make sesquiterpenoids or triterpenoids. So there's two possible engineering strategies. The first one would just be to express the rate limiting enzymes of the DXP pathway, and then overexpress, in our case, a diterpene synthase of interest, in our case, a momelactone 1, or whichever one you desire. But you can also use the cytosolic pathway. The only thing that you would have to do is you would have to change the localization of the diterpene synthases that naturally go to the chloroplast. So you can do this by just removing the N-terminal peptide, that's the localization sequencing tag. And then you also have to overexpress the rate limiting enzyme of the mevalonate pathway with it, which is HMDR. So with this in mind, we tried both strategies to see which one would work better. And we started with taxidine just simply as a proof of concept because it's a very well characterized enzyme, and people have been engineering taxidine for many years. And I kind of want to show you the hypothesis and I guess images, because I think this sometimes helps the clarification of things. So here I'm just showing a plant cell that has been infiltrated with GFP. And this is kind of what the data looks. So you can see these kind of like green spots, I kind of think they look like jigsaw puzzles. So this is a plant cell. So that model that I just showed you as of a rectangle is completely erroneous. Plant cells have a little different shape. But I also want to highlight those like little purple magenta spots. Those would correspond to the chloroplasts. So chloroplasts have chlorophyll, so they have a natural autofluorescence. So with that in mind, whenever we express the native taxidine synthase, that one, as we expect, goes into the chloroplast. But when we express the version, which I'm now calling a cytosolic taxidine synthase, where we remove the N-terminal sequence, we can now see expression of this protein fluorescent construct in the cytosol. And with that in mind, knowing that our enzymes are moving in or are localizing in the right compartments, we were able to show that expressing the pathway in the cytosol resulted in an increase in yields. And more importantly, we could then go on and test the later enzymes in the non-lactam pathway. So I know I said that I just show you stuff about taxidine. Another reason why we started with taxidine is because taxidine is an enzyme that makes the diterpenes. It's a single diterpene synthase that generates the cyclic scaffold. Most diterpene syntheses in plants require a diterpene synthase pair for the scaffold generation. But essentially, we did the same strategy, and we were able to show that through the engineering in the cytosol, we were able to see the product that we were not able to see before, which was really exciting for us. And with the core pathway now set, we went on and tested all the other enzymes that had been characterized, and we mined more data sets for expression data sets that are publicly available for more enzyme candidates. And again, I guess I kind of like gloss over this, but I kind of want to show the way that we test candidates. So what we do is the following. So we do a thing that we like to call combinatorial testing. So we create an average strain with our candidate of interest, in this case, a single enzyme. And then we infiltrate that to the plant. And then we add a second enzyme in a different agar strain, and we combine it in the same liquid solution, and we infiltrate that into the leaf, and so on and so forth. But the advantage of this is that you can add many number of enzyme candidates at once to quickly screen through dozens and dozens of genes, making this a very, very easy platform for quick discovery of biosynthetic candidates on any pathway. And once you have something active, you can just do a simple extraction and test stuff through GCMS or LCMS. And our data, I just kind of want to show that like kind of raw data, but what we typically are looking at are just chromatograms, and we're typically looking for generation, consumption of peaks, and we do a lot of untargeted metabolomics because most of the times sometimes we don't know what product we're expecting. And I guess just jumping kind of to the end, we were able to figure out the full pathway to monomolactones. Specifically, we were able to find an enzyme, CYP76M14, that was not previously characterized. And we were able to show that an enzyme that had been characterized in had been attributed to a different part of the biosynthesis, also had an involvement early on in the biosynthesis, but more importantly, we were able to reconstitute this in a heterologous host. Here are just kind of the same yields showing all the intermediates and how it kind of is linearly moving as we're adding enzyme candidates. But through this strategy, we were able to accumulate actually a lot of monomolactones. So here I'm highlighting the yields in rice and comparing them to the yields that we're getting in benthy or tobacco or in benthamiano. So our yield improvement is tenfold, and we only require 80 plants to get to the milligram scale. And this is great because this is the key point that I wanted to drive, that if you don't have a complete pathway, you're kind of limited to what nature gives you, right? But in our case, we were able to, now that we have milligram quantities of compounds, we kind of just wanted to test the activity of malolactones in vitro assay, sorry, in an in vivo assay with plants. So here I'm kind of showing how the seeds don't germinate with the application of malolactones, which recapitulates the allelopathic activity. And we can also see this in seedlings. So I was showing you just seeds. So it was just seeds that we dumped in a plate previously, but these ones are seedlings that were grown for 10 days. Then we transplanted it to plates with malolactone, and you can see that the roots don't grow anymore. Sorry, the roots stop growing in the presence of the compound. But I think this was just kind of like these assays have been done before, but we wanted to just show that we were able to do them by isolating a molecule from a heterologous host. But now we're interested in taking it one step further. So in this case, we can now answer more questions that we couldn't when you don't have a full pathway. So in this case, I'm showing you like our hypothesis of trying to determine what makes a molecule allelopathic and what moieties are necessary. So our guess is likely that the hemiacetal ring is important for allelopathicity, but we would like, now that we have the system, we can remove enzymes from the pathway and get access to these intermediates and try to replicate these assays. But I think the ultimate goal of what we're envisioning is, I guess, engineering of this pathway into a different crop. So rice is exuding these molecules, but what if we can put these molecules in corn, or we can take them to tomato or some other host, and we can, through synthetic biology, we can engineer temporal control of malolactone production, and perhaps get into an alternative, some alternative methods for agriculture that don't necessarily involve the direct application of a pesticide. So with that, I kind of want to thank you all for inviting me and also for hearing my talk. And this is a picture of my lab. Everyone has been great in my lab, and I really, really want to thank them. And Beth has been great, a great PI, and I want to thank some of my funding sources as well. And I would love to answer some questions if anyone has any. Thank you so much. Yep.
Thank you so much, Ricardo, for taking us on a journey of natural products and plants. So, Anthony, do we have any questions? You're muted.
I have to do it once every session, Anya, right? So yeah, congratulations, Ricardo. Really nice work. So yeah, we have a question from Joshua Whitehead. He says, thanks for the great talk. What is the mechanism of allelopathicity for malolactone, and does overexpression in any way harm the plant producing it?
Yeah, so I guess the first question was the mechanism. So that's another question that we're trying to answer because it's not known. And I think there's two things that you have to think about that question. Is like, what's the mechanism of allelopathicity? So how is the other plant affected? And then I guess the other part of the question is, why is rice not affected by malolactones, right? And we have no answer for either. I can speculate. So for instance, it's possible that like rice, in the sense of why is rice immune, perhaps rice has a mechanism for degrading this molecule, or it can just kind of like compartmentalize it and say the plant vacuole or something like that. But we really don't know. I will say this, that it might just be some I really don't know. It's just speculation, but hopefully we're gonna want to get there. But if you know what, I'm gonna go there. I think it's probably interfering with some sort of hormonal regulation with like gibberellin biosynthesis, which is an important plant molecule, but we have no evidence of that yet. But hopefully we can test that later. Yeah. Oh, and the other one was, why is the plant not immune? Yeah, so I'm using a plant host. Why is it not immune to malolactone? So these assays are only going on for 5 days, and they're going in, I'm infiltrating the leaf. So malolactone is exuded in the roots, so likely the other plant is taking it through the roots. So maybe in the leaf, the target, the ultimate target for malolactone is not in the leaf. Another thing is agrobacterium is also kind of a pathogen. So whenever you inject agro into the leaf, you're also hurting the leaf kind of. So you have to kind of deconvolute those two things. So yeah, it may affect it. I think the easiest experiment would just be to try to infiltrate malolactone directly to the leaf and see if it stops growing. Yeah.
Okay, so we have a second question from Adam. How well could this combinatorial approach be implemented into labs on a chip?
Oh. I think, how could I be implemented on a chip? I think it's hard. So I know I went over here and I'm trying to make it seem like there's n plus infinite number of candidates, right? But I think there's a limit to the number of strains that you can infiltrate into a single leaf, where you can expect all of the genes to come together into the same plant. So you can't just, for instance, create an agro library with every single gene of a plant. So there's a limit there. We don't know what the limit of maximum number of strains is, but it's likely not going to be over like, I don't know, 100 or something like that. So going to the very like high scale might be hard, but I think there might be some way to implement this or make it a little more high throughput. But yeah, I don't want to, I guess I should have mentioned that, like, you can't just go like to something like that. Yeah, it's still a little limiting, but still, like, testing, I guess, 24 candidates at the time, it's still pretty good. Yeah.
Okay, so we have a final question from Garrett Peter Miller. So thanks for the great talk. When comparing yields between plastid and cytosol targeted pathways, did you test any different DXS/HMGR/GGPPS enzymes and see if different versions of the same enzyme, e.g.
Right.
from different species, had different yields?
Right, so not for all of them, but I have done that with GGPP synthase, and I've done that with HMGR. So I've tested different versions, all of them plant versions, and they kind of all work similarly. But of course, I haven't done a comprehensive study. And there's also a few other things that you can do. For instance, like HMGR, you can truncate the enzyme to, because it has like an inhibition kind of loop. So there's more stuff that you can still do towards the engineering of this. So yeah, to short answer, we haven't tested everything, but for the few ones that we've tested, they all seem similar.
Great, thanks very much, Ricardo. And I say congratulations again on a great piece of work. And I'm going to pass back to Anya now to introduce the next speaker.
Thank you.
Yeah, so before we go to the next topic, Ricardo, I actually have a question regarding the plant genome organization, because I'm more familiar with the bacterial gene clusters. Are those natural products, biosynthetic pathways, also encoded by gene clusters? And how easy or how difficult is it to uncover them in plants that have significantly larger genomes than prokaryotes?
Yeah. Am I muted? No, right?
You know.
Okay, so Yeah, so there are some pathways that are organized in biosynthetic clusters. So here's a slide. So monomolactones are localized together in a genomic cluster. You might notice this. What you probably define as a bacterial gene cluster is very different to what plant people define as a gene cluster. A plant gene cluster can be like hundreds of kbs in size. So there's definitely precedent for plant gene clusters, but I would say not all pathways are clustered. It appears that it's as we're discovering more genomes, we're discovering more pathways that are clustered, but not all of them are. And in terms of how hard it is, I think one of the biggest issues with, I guess, the plant genomic organization is that plants accumulate a ton of think about a plant. Plants can just accumulate a ton of perturbations in their in genetic perturbation. So a lot of them are like hexaploids or tetraploids or octoploids. And what makes it really, really hard is that they have regions of their genomes that are highly duplicate. So now that at least we're getting, sequencing is getting better, we're getting longer reads, we're better now able to get more information about gene clusters and things like this. Yeah.